Now, again, the important part of our cell membrane is controlling the inside environment versus the outside environment. And what controls that is the permeability of that membrane. When we start to affect cellular permeability, which disease does, then we start to affect the environments inside and outside and the functions inside and outside. And so when we talk about our cells being semi-permeable, another way to refer to that would be differentially permeable, which is mentioned here, is basically that cell limiting, encouraging, or discouraging something being able to pass. Some things can move easily because they fit a certain criteria, and some things less so. So the cells let some things pass, like water and some nutrients, but it doesn't let other things pass. And that's good, because another example of why we want to keep certain things in a certain place is you look at digestive enzymes in your intestinal tract. Well, your digestive enzymes are there or produced for you to be able to digest proteins, lipids, and carbohydrates. And you, wait a minute, what are we made of? Proteins, lipids, carbohydrates. And so if some of those digestive enzymes leak from your intestinal contents out into your tissues, well, instead of digesting, okay, the Krispy Kreme you ate, you're going to be digesting yourself. And so there's a reason for this permeability and why we want to control it as much as possible, or what's appropriate. Now, this looks somewhat complex, but I think the easiest way to reference this information, and I'll come back to the slide, the easiest way to reference this is those things that can easily pass the cell membrane are those things that are small, neutrally charged, and lipid soluble. Okay, so those things that are small, neutrally charged, and lipid soluble. Now let's look at the graphic, and this should make more sense. So here are a whole bunch of things that are small and neutrally charged. Therefore, they can easily pass. Well, why do they fit that criteria? Well, small, to be able to fit between the molecules. Neutrally charged, because you look at the biggest chunk of the cell membrane, which are the fatty acid tails, which are just carbon, hydrogen, carbon, hydrogen, carbon, hydrogen, carbon. So they are neutrally charged. And so they are like molecules in terms of their charge. They're also, all those fatty acid tails, they're lipids. And so that's why those are the things that pass easily. So here's small and neutrally charged, okay, and to some degree lipid soluble, so they fit. So our ability to have water move, our ability for our ammonia to move, CO2 to move, and oxygen to move. Now notice here's a great big hydrocarbon. Remember, a hydrocarbon is carbon and then totally saturated with hydrogens. It passes because it's neutrally charged and lipid soluble. May not quite fit the small category, but it fits the other two of those other three. Now glucose is big, and it's not completely charged. It's not an ion, but okay, it does have polar covalent bonds, and so it's got a little bit positive, a little bit negative that you remember from those bonds, and so it doesn't pass. And so our sugar doesn't pass. Well, that makes a little sense, because when we eat something and we absorb the sugar into our bloodstream, how do we get into our cells? Well, that's where insulin comes in. And so we'll kind of describe its function later as we talk about glucose transport. Now, notice on the last picture, we've got sodium and potassium and hydrogen. Okay, so here's our smallest element on the periodic table, and it can't pass. But yet over here we have CO2 and NH3 and O2. They can pass, but yet hydrogen can't. Well, why? Well, notice this is a hydrogen ion, and a sodium ion, and a potassium ion, and a calcium ion, and a chloride ion. They are charged, therefore they can't pass. So those things that are, again, small, neutrally charged, lipid soluble. And so we solve, and the question here at the bottom is the cells need some of these things to pass. So how does the cell solve that problem? Well, what we're going to do is we're going to use some of our transmembrane proteins to facilitate that. And so that's where glucose comes in, and its relationship to insulin in terms of transporting those. So essentially, as we kind of take them from the most permeable to the least permeable, so those things that pass easily versus those things that don't pass, we have our gases, our small fat-soluble molecules, and then our charged stuff and our proteins don't pass. So we're going to have to manipulate those. Now, water has some special properties associated with it. We'll talk about how that works a little bit, but it's kind of a special thing that comes into play.